thank you very much for being here. Thank you very much for the invitation. I know so many KLI fellows and I've never been here, so I'm delighted to at last be able to see what KLI is about. So today I'm going to talk about getting Mendel right, but actually I have no problems with Mendel. My problem is that his name is associated with a naive model we teach in schools. And I'm going to talk about teaching in schools because this concerns the majority of people, not the very few that will become scientists, but the vast majority that will be citizens. And following on what Brian said, I think this is a big issue. So here is an overview of my talk. I'm going to briefly present what I call the stereotypical account of Mendelian genetics, and then explain why it distorts both history of science and science itself. And I will end up with some suggestions about what we had better do in schools. So I begin with the first point. Of course, since yesterday we've been talking about Mendel and his famous paper published in 1866. My view of his and my understanding of his is that he was a brilliant experimenter who was adopted after 1900 as the founder of genetics, but he was not really doing genetics. Nevertheless, the stereotypical representations in textbooks usually present the characters that he studied like this, dichotomous characters, either or, something like that. And of course, the famous crosses, the famous ratios, and the laws which are associated with those. And then, if you look at textbooks, and you can find some quotes in this paper here, they're talking about the study of genetics having begun with Mendel, that no one understood Mendel's work, that it contained the paper when published the entire basis of modern genetics, that he studied these either-or characters in detail, and even more than that, that he observed that different alleles were segregated, and that even discovered meiosis. So sometimes things go too far in textbooks, and this is something to be of concern for us. We also have websites like this page on the website of Nature. It's a Nature education section that is not being updated anymore, yet it's still there. And students can read that he hypothesized about particulate matter that is being transmitted to offspring, and that he explained in his notations that there are alleles, factors, elemental, whatever, that is being transmitted from generation to generation. Similar here in Encyclopedia Britannica, Mendel's genes were only hypothetical entities, factors that could be inferred to exist in order to explain his results. And worse than that, popular books like this bestseller, which tell us that Mendel discovered gene in 1864 and then the concept was forgotten. And if he just had stopped experiments there, that was enough because he had already established the foundation of modern genetics, and that he managed to understand and reveal all the properties that what we now call genes have. And worst of all, what I completely dislike is this stereotype of the lonely genius. The lonely monk, actually he was a friar, in the garden doing experiments isolated from his context, but as we know since yesterday, this is far from true. He was a man of his time, working in response to practical problems. And the reason I dislike this is because students are familiar with these kinds of stereotypes. I don't know if you have seen the series, you know, Sheldon Cooper, the genius who is also strange, idiosyncratic, to use a Greek word, and Mendel was not at all like that. So we have representations about what he did, what he discovered, about being a lonely genius, and as I'm going to explain, most of that is quite inaccurate for different reasons. Let us first see what happens with history. There are many translations of Mendel's paper. There was one by Bateson in the early 20th century. There was one by Stern and Sherwood in the 60s. There's a recent one by Abbott and Fairbanks in 2016. But I do like this one, and that's a book I got at the museum 2 days ago, because I already had, of course, the online version, because for people like me who do not read German, it explains the use of German terms. And that's a very, very, very useful tool to understand. And this was done by two historians who are experts on this, especially Stefan Miller-Wille, but also Kirsten Hall. They have worked on that for years. So if you actually read what Mendel's aim was, as he wrote, was to observe these changes for two differing traits respectively and to investigate the law according to which they occur in the successive generations, and that was the task of the experiment. So he's talking about trait pairs, and he makes a distinction between them, and he's talking throughout the paper about characters. And he explains that there is a segregation of characters. At some point, they are together, and he's using the term hybrid traits, but it refers to a character being a hybrid, not the traits of hybrids. And the most important thing, what is dominating or dominant or recessive are the characters. He's not talking about alleles, elementor, factor, or whatever. Staffan and Kirsten explain exactly how they understand the use of this term in the paper. It is always the characters that are transmitted, and this is what he's interested in. And as Greg explained yesterday, that was mostly about the hybrids being constant or not in response to practical questions of the time. Most interestingly, when he explains his results, he comes up with this ratio, which is not what we find in textbooks. He is talking about a developmental series consisting of 9 members, and he describes the features of these members. So the results are these, and of course anyone familiar with genetics will figure out a 9:3 to 3:1 ratio. But what Mendel presents here is not that. He presents 9 different classes of phenotypes which give this ratio, 1 to 1 to 1 to 1, 2 to 2 to 2 to 4. Whereas what we are teaching in genetics today is a bit different, or actually quite different. We're using a ratio among phenotypes based on the alleles that the individuals are supposed to carry. And it was Bob Olby who clearly showed this difference in his classic 1979 paper, and later that was included as an appendix in his book. He makes this table, that's not my idea, it's his, and he shows what Mendel did and how he came up with the results I just presented, and what we do in today's genetics. And I think the difference is obvious, and I think from this is clear, at least I am convinced, that Mendel was not talking about alleles, factors, or whatever hereditary particles being inherited. He was looking clearly at phenotypes and traits. So, in other words, we can see that whereas in Mendel's terminology we have A as the dominant trait that remained constant through generations, A capital, and the small a, that was the recessive trait, and big A, small a was the hybrid form that somehow exhibited the features of the dominant one, but also had in a latent form the recessive. This is very different from what we do, talking about genotypes, homozygotes, the 2 homozygotes here, and the heterozygote. So in short, his own notations are quite different from the notations that we use today, and my understanding is that they're different because he was doing a different thing. He was studying the transmission of traits. So if we read his paper closely, here is what we can find. He intended to study hybridization in particular and not heredity in general because of the practical interests of his time, and I will say a bit more about this in a moment. He studied how traits were passed on from one generation to the next one, and nowhere did he write anything about hereditary particles. And the most interesting, he was able to observe the segregation and independent assortment of traits. That's clear in his paper, the 2 laws that we attribute to him, but he's talking about traits, but he does not conclude anything about the segregation and independent assortment of alleles. And that's a big difference, in my view. Now, turning to the nature of science part, students have this view intuitively that, this is how science is done. Some weird individuals working on their own, whereas, as you know, the work is done in scientific communities. Of course, it's not that bad all the time, but scientific knowledge is produced by communities. And this is what happened in Mendel's time. So, while Mendel was doing his work, there was a whole community of people interacting with one another, starting with Charles Darwin, who placed the questions of heredity at the center of biological thought, exactly because he could not answer them. And then we had other people like Herbert Spencer, who was the first to talk about particles in 1854, Francis Galton, who somehow refuted Darwin's pangenesis, and then Brooks, Weismann, Hugo de Vries, and others. I'm just mentioning a few. And the only one who came in touch with Mendel was Carl von Langelier, the Swiss naturalist, who had a very good correspondence with Mendel, even though we do not know exactly what he told him, who was criticized in 1965 at the centennial of Mendel for not understanding Mendel's work, and who nevertheless seems to have discussed fruitfully and constructively Mendel's results. Now, if we look at Mendel himself, he seems to have been working in the hybridist tradition ever since Linnaeus. And as Stefan and Vítězslav Orel explained in detail in this very interesting paper, if you look at exactly what Carl Linnaeus wrote, like Kölreuter and Gärtner, Linnaeus rather superficially thought that hybridization can produce new species. Kölreuter and Gärtner thought this is not perhaps the case, and Mendel perhaps was trying to answer this question with his paper. So he was working in a tradition of hybridists with specific aims that eventually led to a focus on the inheritance of individual traits. Then in 1900 we have the rediscovery. This is presenting as a moment where everybody arrives at the same results and they think of Mendel, and then Mendel is put back on the scene. But as Augustin Branigan explained again in 1979, I don't know if this is a coincidence, and then in his book, this was a case of reification. So what happens is that we have a paper in French by Hugo de Vries that does not mention Mendel. Correns, who was a student of Nageli and probably know about Mendel, becomes frustrated, and he writes a paper, a German paper in this journal, where he mentions Mendel with this title, Gregor Mendel's Results on Hybridization. And according to Branigan, is this title that perhaps was used to resolve a priority dispute between de Vries and Correns that puts Mendel back to the scene. So, why Hugo de Vries did not mention Mendel? Historians disagree. I think that the view that he did not think of Mendel's results as important given his own views is the correct answer. He does not seem to have tried to mask, the importance of Mendel's work. And so when Correns brings him back, we have the reification of Mendel. Not that his work was not important. It was important, but it was not the work on heredity in the way that it was later co-opted by people. Now, what had happened in the intervening time, in this 34 years from 1866 to 1900s? We have Francis Galton, who had developed a framework of discontinuous variation, and who speculated about the germline and germline inheritance, something that August Weismann established, mostly influenced by the advancement of cytology of his time. So what happens is that Mendel's paper was read in a new light, and whereas he does not have a theory of heredity, his brilliant experimental approach is suddenly found very useful in the new framework. And this is where the reification brings in something useful. He provides people, those that will soon be called geneticists, with a very useful experimental approach to design model systems and do their work. Another important issue is that Mendel's paper was cited and discussed in various publications. This is a 1968 paper by Olby, and there's another one in German. Overall, I think there are more than 22 references to Mendel's work before 1900. So it's not the case that it was unknown. Whether Darwin read it or not, it doesn't make a difference. Greg has just written a chapter for a book I'm editing where he shows that even if Darwin had read the book, nothing might have changed. Perhaps Galton could have appreciated that, but we will never learn. So I very much agree with the conclusion that Olby makes in his paper, in his book, that if we think of someone as being a Mendelian because he subscribes explicitly to the existence of a finite number of hereditary elements, then Mendel was no Mendelian. But if we think of someone as Mendelian who treats hereditary transmission in terms of independent characteristics, character pairs, and the statistical relations of hybrid progeny as approximations to the combinatorial series, then Mendel was a Mendelian. And I think that it is in this sense that we can say that Mendel's work was not relevant for the study of heredity before 1900, but once the framework of hardware heredity was there and the understandings of cytology were there, then it made perfect sense to people. So, all in all, the problem here is that we have someone who works as a person of his time in the context in which he lived, doing very important work. There are newspapers, local newspapers, referring to Mendel's work and to Mendel's presentation, which shows that it had an impact, and nobody questions that. But this is different from developing a theory of heredity. Now, the most interesting stuff, and what I think is even more important, why the stereotypical account of Mendelian genetics taught in schools, I repeat, distorts science. So, this is what we teach, that there are genes for traits, genes for yellow color or green color, genes for wrinkled or round seeds, etc., etc. There are at least two main problems with this. The first problem is that traits are presenting as being dichotomous, even though they're usually not. And in this way, natural variation, the variation that exists in nature, is ignored. And this was pointed out already in 1902. Greg explained that yesterday. We have a continuum from greenish yellow to yellowish green. It's not either or. And we shouldn't forget that Mendel spent 2 years to arrive at pure characters, what we would call homozygotes. So what Mendel practically did is that he eliminated any natural variation. And of course, this is legitimate to do in a research setting to create a model system, but this doesn't mean that this is how inheritance works in general. And there is more to read in Greg's book. I urge you to read it. It's long, about 600 pages. It took me a lot of time to read it, but it's magnificent because it not only sheds light to this history, it will also make you better understand how we should study history. Another example, typical example, we have already seen that. Eye color. Already in 1909, there were references that it is possible for two parents with blue eyes to have a child with brown eyes, in contrast to what we teach in schools. In schools, we say that brown is dominant, blue is recessive, therefore, this is how things work. Another problem is that this ignores all this variation in eye color. Eyes are not green or blue or brown or whatever. There's an enormous variation. And this is something that we need to show to students. Of course, we can create models with at least 2 genes that can explain things, but even this that I'm showing to you here from my book is an oversimplification. But at least it's a bit better than the overly simplistic thing that we saw before. So, one point is that traits are not dichotomous. Things are more complicated than that, and this is something that students should appreciate. The second point is that the relation between traits and genes is presenting as being one-to-one, even though we know today that it's many-to-many. And single genes are difference makers. We had this discussion yesterday. They do not, on their own, determine traits. And here is an example of what we teach. We teach that there is an allele for red eye in Drosophila, and there's an allele for white eye in Drosophila. But this is entirely wrong. And I'm going to explain why, but I'm going to quote some scientists here. So, if you know where this comes from, please don't speak. But if you do not know, I challenge you to guess when this was written. It says that red eye color in Drosophila, for example, must be due to a large number of factors. For as many as 25 mutations for eye color at different loci have already come to light. So, one can therefore easily imagine that when one of these 25 factors changes, a different result is produced, as in pink eyes, vermilion eyes, or whatever, but we need all of the 25 factors together to have eyes and eye color. Can you guess? This can sound, of course, a bit modern. Any guess by someone? Okay, so this is 1915, Thomas Hunt Morgan. And we don't teach that in schools, even today. So he came from embryology. He was very sensitive, whatever has to do with development. And he had a brilliant group of people working on inheritance. They did not adopt the term gene right from the start, but they clearly showed how we could imagine genes or factors as beads on a string on chromosomes, as distinct segment of chromosomes. Most famously, Sturtevant made these maps of genes on chromosomes and things like that. And what they showed is that, here is a representation, whereas we need 25 genes to have the eye color, it is of course possible that if one changes, there will be a different color. But this is not the gene that causes the color. This is the difference maker. And that's a very, very, very important concept for understanding heredity. And this is what he wrote in his 1934, he got the Nobel Prize in 1933. The lecture was published a bit later. In the earlier days of genetics, it was customary to speak of unit characters in heredity because certain contrasted characters, rather clearly defined, furnish the data for the Mendelian ratios. Certain students of genetics infer that the Mendelian units responsible for the selected character were genes reducing only a single effect. This was careless logic. And I assume you understand what goes on. He insists that it's clear that genes have many effects. There may be a main effect, but there are also other effects. And this is 1934. And we still don't teach that in schools. What happened, and this was lost, my understanding is that genes became molecular, reductionism prevailed, but that's a different lecture. We can discuss this later. Another important issue for school. This is a paper, there are others, reviewing what goes on with Mendel's genes, as we call them. And it's clear that Mendel's genes are not about shape or color. These are genes involved in metabolism, starch metabolism, chlorophyll metabolism. And when they lose their function, we have different phenotypes. Exactly what Morgan said. We have genes being difference makers, not genes for factors. And it is the problem that I'm arguing against is the idea that their single genes determine traits. And you will see in a moment why this is bad. So that's the problem. And it's a problem that Mendel's name is also associated with that. I'm sure he understood development a lot better than that. The idea that a single gene determines a single trait. And Amir showed yesterday how this naive understanding can be abused in very different ways, and that's exactly what also Brian is arguing against. And so, what we see in the media is that there are genes found for this or that disease, and people go crazy with that, whereas what we actually have is associations. And I call this concept in my book the associated gene because this is what people misinterpret. They misinterpret association as causation. And whenever they read headlines like this, they think that a gene was found that does that produces that disease, even though this is not what happens in genome-wide association studies. We compare groups trying to find correlations between people with a disease and healthy people. Now, healthy people are hard to define because they may be on the progress of developing the disease, but that's a different story. And the most important issue is that insofar as we do not know the mechanism, how this association works, so do the genes cause the condition? Is one gene regulating the other? Or perhaps both regulate the expression of a different gene? Until the moment that we understand the underlying biological mechanism, the association is just a statistical thing that is important, but we do not understand what is going on. So we have pleiotropic genes and polygenic traits, and this is something that we need to explain to students. How I will show you some suggestions in a moment. But we need to give them somehow a sense of the complexity. There are monogenic diseases, which are rare, and there we can make the inference from the disease to the gene, but people often think that this works the other way around. You find a gene in a genetic test by 23andMe, and then you know what is going to happen to you, but it's not as simple as that. So, to conclude, okay, I explained the problem. What are we going to do with teaching in schools? Can we teach GWAS? Can we teach molecular biology or whatever? No, of course. The most important thing is that we need to figure out what students think. And there are many people involved in science teaching and communication that think that making things simple is the best way to go. And they're entirely wrong. The first step in teaching and science communication is to figure out what people think. And what you have to do is to address what they think. So this is a review we did a few years ago that shows the sources of misconceptions about the nature of genetic material, the role of genes, and the nature and potential of genetic technologies. Textbooks are responsible for all of them. Teachers for some of them, the media for some of them, and genetic essentialism that Brian explained for this issue. So we know the sources of the misunderstandings, and we have to figure out which kinds of misunderstandings our students have in order to proceed. Furthermore, we need to understand, after we see in detail what is going on, to what they're exposed. They're exposed on media messages like, if you have a happy relationship, it may be due to your genes. So there's nothing you can do about that. And if you don't have a happy relationship, again, it may be due to your genes, because there's a fidelity gene or whatever. And if you have no relationship at all, don't worry, because we're here for you. Science will find a way to get you the good partner based on DNA. These are actually solved. And this is what people find. And unfortunately, the naive understanding of Mendelian genetics makes this very simple to grasp and understand. So, there are studies about whether to teach Mendel before or after. Some show, like this one here, that doing molecular first is better. Others show that comparing developmental perspectives and Mendelian genetics makes a difference. This is a study by Greg. Others found no difference, but we only have a few studies and we can make no inferences. So I wrote this chapter indeed, should we give peace a chance? And my answer is no, because I think it does more harm than good the way we do this. And what we should do, we should introduce development. I think that, paraphrasing Dobzhansky, heredity makes sense only in the context of development. And I like this illustration that shows how we advanced from simplistic understanding over the 20th century to understanding the importance of development and environment in the production of traits. So, what we need to replace is this simplistic understanding. Currently, we teach that once you have the recipe, this is the DNA, you will have the meal. This is the phenotype. And this book again, obviously I like it a lot. He explains that the SRY gene is the main determinant of sex, whereas we know that sex determination is a matter of many proteins, and of course mutations in SRY can make a difference, but it's not the only gene that determines sex or maleness or whatever. So I think we need to find ways to introduce the importance of context. So my idea is this. We explain to students that once we have DNA, we may have two different developmental systems that are using this DNA, a famous Ceph on the left and another one on the right, and depending on the developmental system, we can have different developmental outcomes. And I have empirical evidence for both of them. So I think that this is when heredity will make more sense to students, rather than the simplistic understanding from gene to phenotype. Now, going back to Mendel and Nature of Science, we did an analysis of a few textbooks many years ago, and we found many instances of Nature of Science. Christian explained that it's important to be explicit because implicit doesn't lead us anywhere. All the instances were implicit. So we lose the opportunity with Mendel in textbooks to teach about how science is done. This is the case for aspects of nature of science, or nature of scientific knowledge, and another one for the nature of scientific inquiry, only 3 explicit instances. So, to conclude, I think that Mendel was an ingenious plant breeder of his time, not a lonely genius ahead of his time who understood heredity. And the Mendelian genetics as currently taught provide an oversimplified and over inaccurate explanations for hereditary phenomena. So what we need to do, I think that perhaps we should drop Mendelian genetics altogether from genetics curricula in schools. Why? Because we leave students with that. If we integrate this with developmental explanations, then things might be different. At the same time, we should be teaching about Mendel, but we should use his very interesting story and well-studied story to give lessons about how science is done, that it's a matter of communities, that things are not linear in how advancement is made. And I think that Mendel's story can provide useful lessons for nature of science, but not for today's genetics. And actually, I repeat, I think his name is unfairly associated with the simplistic model I just described. And with that, I would like to thank you very much for your attention.